is you've shown these different data with the different doses of radiotherapy. For a fact, I've done studies where I've shown that giving fractionated therapy is advantageous because it activates anti-tumor immunity. Is that something that has been studied in your study? Have you looked at the anti-tumor immunity? My second question is, you've shown that the software is capable to see calcium, and we know one of the immunological signals is when catalytically release the calcium for cancer cells. Is that something that is feasible through your software to kind of live assessing immune response, anti-tumor immune response using this software with different radiotherapy doses? And my last question to global partnership was like one of the things that, because I'm working with the Gabonese government, and what we're trying to implement is workforce training. Like you just mentioned, there are not many people. And for a fact, here in the state of Massachusetts, I'm part of a program that trains the next generation of biotech entrepreneurs. And what we do, we work with biotech companies that sponsor co-op or accelerator program for the mobility of the students. Is that something that is envisioned globally, where like different countries can collaborate and maybe industry stakeholder can contribute, not necessarily to provide the co-op, but to kind of provide mobility of students coming into an institution from one country into another to get training on certain machines? So that would be my questions. Thank you.
Right. I'll answer the first two questions. The first question was about immune responses induced in cells. Absolutely, that is something that we care about modeling. We have modeled that into the simulation. However, the real question is how much of an immune response should you model in response to a certain type of treatment with a certain cell or a cell line. So we have all of the calculation parameters built in there. What we really need is guidance from the medical community on what should be modeled. This is a simulation, right? It isn't magically making up answers for you. It is calculating things that are extremely difficult otherwise. It is simplifying the calculations and coming up with an answer based on information that it is providing. So yes, we have modeled these factors. However, we need to make sure that the value provided to the factor at the time the simulation is being run is correct. And same thing, in fact, with the calcium ions. It's simply about modeling specific pathways. If we have modeled a pathway in which calcium ions are being injected, emitted, and tracked, we can do that. One of the things that we have done is evaluated the presence of free radicals in response to treatments. So yes, we have done that, but again, the same point comes in. I don't know yet how much free radical concentration you would start to see if I treated patient A versus B versus C.
I can try to address your second question, which is about training and coordination. So I think that's something that maybe can come out of this, because for us, we do a lot of work in navigation, and now what we're trying to do is coordinate those efforts, because they're happening in multiple different places. They're different training curricula, trying to set some standard consensus so that we can move forward with one and share. So I think that's the thing, is that there's so many different training programs that are happening, lots of twinning that's happening. How can we think about, and there's lots of different training curricula, we as an organization have been thinking about the non-oncologists, because there's lots of task shifting happening in LMSCs, and how do we develop digital training curriculum together for those professionals. But again, I think there's so many programs happening. Can we leverage technology to be able to even understand who's doing what training curriculums are happening? Can we develop something together, coordinate? So, I mean, again, I think that's a question that I'm hoping technology can help us answer. I don't know if you want to add to that or others.
Thank you.
Right. Oh, yeah.
Hello, I just have are you finished with what you had? I just have another question. I don't want to.
Yeah, and please introduce yourself also.
Oh, my name is Bilinga Tendwa from South Africa, and I want to ask Dr. Kapadia about. I have a question concerning enhanced sampling and molecular dynamic. This is a topic that is very close to my heart, because very passionate about drug discovery and drug repurposing. So I wanted to ask, in your molecular dynamic simulation, have you looked at drugs like cancer drugs and how they affect the body, and how mutation will affect those drugs in terms of metabolism, and how you could actually bypass those mutations, maybe by drug repurposing or discovering other drugs that could enhance the work of the drugs that are being used at the moment. And due to mutation, they cannot work properly.
One of the challenges with the drug discovery field is the speed of computing. It takes a long time to evaluate all of these possible molecules and how they will interact with the body. One of the things that we are doing, I didn't include that in the presentation here, we are developing a GPU accelerated platform that will let you do exactly what you said. We are testing that in the context of cisplatin interacting with specific receptors. So that is something that's underway. And you're absolutely right about the applications. Once you have that, you can test all of the pathways with which a drug will interact with a certain target. Yes, we have the ability to model the mutations. We will simply model a different target. And then you can evaluate what other molecules are possible that can then target that particular receptor. More importantly, this platform that I showed you, it doesn't tell you only what happens once you get your molecule to the target. It will also let you evaluate how do you get it there. So in that sense, it is a more comprehensive, a more holistic simulation of the human body.
Great, thank you so much. I'm going to have Dr. Uddin join us. He gave some really inspiring and also very humorous remarks at dinner last night. So
thank you. Thank you.
He is with the Oncology Club of Bangladesh as well. So please introduce yourself and then.
Good morning, everyone. May you allow me to use my very important hat now. My name is Brian Chitsonge. I serve as the Pan-African Parliament, which is the legislative body of the African Union Food Security Ambassador for the continent. I just want to say thank you so much for making the dream very simple, and I think it's something that we should actually pitch it to the heads of states. If they understand it the way you've made it, I'm sure all of them, they'll understand, and you simplified it to a point that they will all understand it, and that way they can allocate a budget for what's needed. Without their buy-in, we can sit and we can have as many symposiums, many summits, many everything. There's no point, because most of the time is lack of understanding. I sit with a lot of them and they do not understand. You even talk about simple things, they preach about climate change, carbon exchanges, carbon sink, carbon sequestration, and they're like, what does that mean on the side, you see? Because they don't understand, but the more they understand it, the better. So I think in my capacity as your ambassador, I'm going to see how we can actually get it to the heads of state, whether get them at the African Union where they're all together, or visit them country by country so that we can get them to understand, because the minute you get one to understand, they can take it to the African Union and then discuss it amongst themselves, because this is amazing. I mean, this is technology at its best. Let them understand it. Thank you so much. Very informative sessions. Thank you, GHC, for putting this together. Thank you.
Well, thank you for that.
And I think we can all say that we're all at the ready to do what you just said. If any of us have the opportunity to help with that process, it'll go a very long way. We're all ready to get on planes and do what is needed. Thank you.
Yeah, and I think the Rewire framework is perfect, right? It's a perfect example, but yeah, let's talk more. I think that's great. Please.
Just a question to thank you so much for your speech. But I don't know, in my country, in the first time, patients use a traditional medicine. I know in the traditional medicine we have substance, we can clean free radicals. I don't know what your experience is. So maybe the future ways again to have solution against cancer. Traditional medicine and cancer in Africa.
Potentially. Now I can't. My background is in biomedical engineering and physics and computer science, so I come at the medical field from that perspective. My goal over here is to create this platform where anyone who wants to build on it should be able to do so. So I haven't looked at traditional medicine applications myself, but there's no reason why it couldn't be modeled using a platform like that.
Thank you so much for this talk. My name is Andres Diaz. I'm an MD PhD student from the University of Arizona. I've been coming to the summit for some time, and every time it's just awesome. So I'm really glad to be here. I just have two quick questions. My first question revolves around diagnostics. We've discussed the role of prevention and also the role of increased access. But what I've seen in my experience a lot is diagnostics limiting what is already accessible. In pediatric cancer, methotrexate is staple drug especially for liquid malignancies. But methotrexate monitoring is critical for leucovorin administration. And in some low resource settings, without plasma MTX monitoring, you follow a schedule, and that schedule can lead to suboptimal dose of methotrexate, which leads to worse outcomes. So I was hoping if you guys could speak about the role of improving diagnostics to already leverage the tools and the drugs that are available in low resource settings. And then my second question was a more technical question about your model. We are working on an enzyme that leads to chromatin remodeling. It's called CDK5. It phosphorylates histones at certain locations, and that phosphorylation is leading to chromatin opening. But we're having difficulty doing ATAC-seq and cut-and-run sequencing to see what's being opened up and transcribed in pancreatic neuroendocrine and neuroblastoma. And I was wondering to what degree providing you with post-translational modifications on chromatin structures could lead to a model that could tell us what is being opened up through the model.
We need to talk more about that. Can't come up with an answer.
Because that epigenetic change is so physical.
Yeah.
There's a very mechanical aspect to that.
Right, when you think of a model like this, breaking it down, think about there's a starting point, there's an ending point, and there's what happens in between. You can work with any of these. If we didn't have what happens in between an end point, and we only had a starting point, that is fine. We can model a cell with a chromatin in the starting structure and then determine what goes on. If you had the start and the end, we can use that as a differentiating variable and compute what happens to a cell where you are at the end. If we know what happens in between, we can model it. If we don't know, but you have guesses, we can model all of the different pathways and then let the simulation calculate probabilities and tell you what is possible in what scenario. So again, the simulation is not going to magically give you answers. It is going to help you manage the calculations that go into creating a simple solution from a complex set of variables.
So the more variables we provide, the better the model will be. Because
Exactly.
we have interacting protein binding targets that we can review. But after that, we don't know what's happening. So, anyways, thank you.
Does someone want to address the first part of the question?
I could talk a little bit about the diagnostic piece. I think that there's been a lot of discussion, particularly in Africa, about pathology and laboratory medicine, how to develop those capacities. And there's been a lot of work that's gone on. USCAP and other groups have had initiatives that both train up individuals, pathologists and laboratory medicine experts, also histotechs, cytotechs, people that will support these activities because the number of pathologists in low resource settings is so low. It's very difficult to bring up the number quickly, as we've seen. And in addition, there's a lot of opportunity and quite a few successes in telemedicine, telepathology, where images, pathology images can be rapidly read, and tumor boards, molecular tumor boards, and others can be brought up to help train people on the ground. So there's a lot of activity in that space, certainly not enough to address the question you raised, but it's certainly, we have good models for how it could happen if it could be brought to scale.
The other thing I just wanted to add to that, having treated a lot of leukemia patients in Africa, is that we take for granted all the supportive care that happens to support the toxicities, and the gains and outcomes have been because we've been able to intensify therapy with complementary improvement in supportive care. So that has to go together. So I think that's another really important aspect. You can go to the next question.
Hi, my name is Tim Xiao. I'm a consultant with a firm called Avanticor. Previously having experience working at USAID, NIH, and Astro. So I have a question for Dr. Kapadia. For the digital twin topic you mentioned, when we try to disseminate this beyond the US, then the medical imaging component regarding the international collection of medical imaging will have very different devices and different standards, different routine of annotation, etc. What will be a good way to harmonize those images to build a consistent digital twin? Secondly, as we know that quantum computing is along the line, maybe in the next 5 years or so, we might have a computable model right now. So aside from the molecular dynamics modeling for pharmaceutical compound you have already mentioned, what will be some other components that you think will be the first use case for quantum computing on the digital twin data? Thank you.
For the first part of the question, the images that we created, 10,000 CT scans, they were made using a certain scanner. So we had to build, Duke University has a project where they have built a virtual CT simulator that takes into account the manufacturer and what protocol you want to use, what sort of anti-scatter grid is in there. So these are, some of these simulations are extremely manufacturer specific. You could take this XCAT family and then scan them with 5 different manufacturers, wherever they are. You could use the specific protocols that are used in a rural hospital in Kenya versus a modern hospital in Congo or wherever else. The platform lets you do all of that. And your second question was about quantum computing. Quantum in biology and health is such a chicken and egg problem, right? There's a lot of promise, but we haven't been able to demonstrate that promise because we haven't had computing that can truly prove that application. Some things that I see possible is if you think of the whole field of quantum sensing, you can look at all of these effects in molecular dynamics, in biochemistry. They go through a series of interactions, many of which will have interim states that we have not been able to see or detect yet. If you couple quantum sensing with a pathway like that, combined with quantum computing, now you might be able to look at all of these interim states, which then might hold a key to treating a disease in a different way. We're no longer focusing on the start and the end point, but focusing on something in between that just breaks the pathway. That is my hope for quantum.
So I think we'll take 2 or 3 more questions before we break, so if there was additional questions.
Hi everyone, my name is Doris Kezia and I'm a first year PhD student in medical physics at Hopkins. I'm originally from Cameroon. So my question is directed to you, Dr. Anuj. It's about the digital twin. So it's a really wonderful idea, but it also sounds like really expensive. So like how would, let's say my grandma in Cameroon, like how can she get access to this, and like how long is it going to take for this to like get back there, you know?
Absolutely, it is expensive right now. We don't have all of the capabilities. Fortunately, we have the computing, we have the funding and the resources to make this a reality. Like I said earlier, again, we cannot be unrealistic about this. I do not expect for a second that every single country, every single user is going to have the type of supercomputing facilities that have needed to make this a reality today. The goal is to bring in AI so that eventually this sort of model will run on your cell phone. Imagine if each of us could carry a digital twin of ourselves on our phones. You go to your doctor, you plug in your phone, and suddenly the doctor has the information you need, you're done with your visit, you take your phone out and you walk away. So these are ways in which you have to think about how to disseminate the technology in a way that is realistic. Even when you go to Cameroon, my grandma in India had a phone several years ago, which I wouldn't have imagined possible when I was growing up. So as these technologies develop, we keep an eye on how other things, other supporting technologies are also developing so that we can leverage them.
Maybe we'll take one last question. I know there's a lot more, but please.
That's why it's not a question. I was just going to address what she's just mentioned about Cameroon. That's why it's important that we see the heads of state. Maybe they can do it region by region. In the southern region, they have X number of machines there, then start region on a region or 2 or 3 countries together, because we've got 55 countries on the continent. So they could most probably do like partnerships where we can see how we can lobby. So that's why the lobbying is very, very important. Because of course it's important. It's very expensive, so it's not every country that will be able to afford, because you've got some small countries that have only got a population of like 2 million, like Botswana and those countries. Then you've got the big giants like your big brother and sister, South Africa and Nigeria, that can most probably afford it. And then also to lobby with our billionaires, the big boys. Let's go and lobby with those as well. So I'm happy to spearhead that initiative.
If I could just add one note about digital twins. The way you presented it, of course, is a goal of thinking about how it can be used in medical care and what, the doctor will have that. I think one of the things we've been talking about in my field in epidemiology and genetics is to use it for research, which, I think maybe was part of what you were saying, but, they're using these novel technologies to improve the way we gain knowledge, generate knowledge about situations in diverse places in Africa, wherever it might be, will push forward what we have to use in maybe more traditional ways for prevention, early detection, or treatment. So there are a lot of different levels at which digital twins can be used, and I don't think that the only one is necessarily the doctor's office, which is a great dream, but there's a lot in between.
Yes.
Yeah.
All right, I think we'll take a break till 11, and then feel free, there'll be a lot of additional opportunities to ask questions. Thank you.